All right, welcome to today's explainer. We are diving into an absolute lifesaver of a bioinformatics tool called TissueEnrich. Look, if you spend your days completely drowning in massive genomic datasets, just trying to decipher exactly where and how specific genes operate across an organism, well, you already know how maddening that data can be. It's like finding a needle in a haystack. But today, we're going to break down exactly how TissueEnrich pulls you out of that noise and makes sense of the whole shebang.
Okay, let's dive right into this. We all know standard RNA-seq databases are undeniably incredible resources, right? I mean, they've mapped out adult human and mouse tissues in stunning detail. But what happens when your research goes beyond them? Seriously, what do you do when you're studying an obscure tissue, maybe a highly specific developmental stage, or an embryonic condition that simply hasn't been cataloged in those major adult databases? How do you calculate tissue-specific gene enrichment then? Well, that exact bottleneck is what we're going to solve right now.
So here's our roadmap for today's explainer. First, welcome to TissueEnrich. Second, the standard reference libraries. Third, defining tissue-specific genes. Fourth, unleashing custom expression datasets. And finally, number 5, interpreting your results. Let's get to it.
All right, Section 1. Welcome to TissueEnrich. So at its core, TissueEnrich basically acts as a highly specialized calculator. Its primary job is to calculate the enrichment of tissue-specific genes within a set of input genes that you provide. Imagine you've just run a massive RNA-seq experiment, right? And you've isolated a set of highly expressed genes. You bring that list of input genes to TissueEnrich, and it goes to work figuring out which specific tissues those genes are heavily associated with. Honestly, it's kind of like a biological dating app where your input genes are just looking for their perfect tissue match.
Moving on to Section 2, the standard reference libraries.
Now, what's really interesting about this is that TissueEnrich comes preloaded with these incredibly robust default dictionaries to compare your genes against. For instance, we've got the Human Protein Atlas, or HPA, covering 35 human tissues. We have the GTEx database covering 29 human tissues. And for you animal model researchers, we have Mouse ENCODE covering 17 mouse tissues. And here's a crucial detail. To ensure your calculations are absolutely statistically sound, TissueEnrich only uses tissues from these datasets that have at least 2 biological replicates. That strict inclusion criteria guarantees that your baseline data is rock solid.
Which brings us to Section 3, defining tissue-specific genes.
Okay, the absolute crucial metric for the algorithm here is the number 5. For a gene to actually be classified as highly specific to a tissue, the algorithm dictates that it needs to have at least a 5-fold higher expression level in that particular tissue compared to all the others. So it's not enough for a gene to just be hanging around. It has to be overwhelmingly active. 5-fold is the magic number.
This builds out a really neat classification system based on the algorithm from the Human Protein Atlas, essentially sorting genes into one of 6 buckets. First up is not expressed. Second is tissue-enriched, and this is that golden 5-fold higher expression in a single tissue compared to everything else. Third is group-enriched, meaning it has that 5-fold higher expression in a small group of, say, 2 to 7 tissues. Fourth is tissue-enhanced, where the gene has 5-fold higher expression compared to the average levels of all other tissues. Fifth is expressed in all. And finally, the 6th bucket is mixed. So when we talk about finding tissue-specific genes, we're really hunting for the VIPs here, buckets 2, 3, and 4. All right, Section 4, unleashing custom expression datasets. This is where things get really exciting.
Let's compare the traditional method of relying on default databases with the advanced method made possible by the TissueEnrich Bioconductor R package. With this package, you aren't stuck relying on preloaded dictionaries anymore. Instead, you get to write your own. You literally input both your genes of interest and your very own custom expression dataset.
I cannot stress enough what a massive, game-changing capability this is for computational biologists. Think about it. If you're studying embryonic development, let's say a specific mouse placenta sample isolated on embryonic day 14.5, that unique time point just isn't going to be accurately represented in a standard adult database. But the R package lets you feed in your own specialized RNA-seq expression data to define what is tissue-specific for your exact, unique biological context. Then it calculates the gene enrichments using your custom reference set. It completely unlocks datasets that were previously unmappable. It's a total game changer.
And this brilliantly illustrates what's happening underneath the hood. Whether you're using default or custom datasets, TissueEnrich relies on the hypergeometric test. Basically, this is the core statistical engine used to determine if the tissue-specific genes you just defined are appearing in your input gene set at a rate significantly higher than you would expect by pure random chance.
Section 5. Interpreting your results. Let's bring it all home.
Once the math is done, here are 3 actionable steps you should always follow. Step 1 is simple but so crucial. Verify the expected tissue is actually present in the dataset you're using. I mean, if you're analyzing a sample list of trophic derm-specific genes expecting to see placenta enrichment, you absolutely cannot use the GTEx dataset. Why? Because GTEx doesn't even include placenta data. You have to use the HPA data instead. Okay, step 2. Set your adjusted p-value threshold to 0.01 for selecting enriched tissues. And step 3, whenever possible, cross-reference your results across all available datasets for maximum confidence.
Now, let me share a bit of a secret weapon for your data analysis, especially when you're using the custom capabilities of the R package. TissueEnrich allows you to provide a custom background gene list. If you skip this, well, the software just defaults to using all the genes in the dataset as the background. But by providing a specific background set and making sure it includes all the genes from your input set, you allow the algorithm to perform far more accurate Benjamini and Hochberg p-value corrections for multiple hypothesis testing. It just refines your statistics beautifully.
So, we've covered a lot of ground today, from default dictionaries to the hyper-flexible capabilities of the TissueEnrich R package. And I want to leave you with this question. What unmapped datasets do you currently have sitting on your hard drive right now? What unique, obscure developmental stages or tissue types could you finally unlock by defining your own custom reference sets? The tools are ready to be downloaded from Bioconductor right now. Thank you so much for joining me for today's explainer, and I hope this helps you solve your next great biological puzzle.